All right, these are your notes for Chapter 10, Control of Gene Expression. Gene control is necessary for individual cells to respond to changes in their extracellular environment. A typical cell uses only about 10% of its genes at one time. For example, most cells express genes for glycolysis, but only in immature red blood cells is there a need to express genes that encode for globin to make hemoglobin. It is crucial for proper development of complex multicellular eukaryotes to have gene control. So this is, again, gene control. Basically what you're thinking of is genes are going to be turned on or off during their embryonic development to make different types of cells. Okay, and genes can be switched on or off. The expression can be speeded up or slowed down by molecules and processes that operate at each step. So if you think about it, you have transcription and translation, and you have how DNA is even wound around in its chromosomes. So the first thing we're going to talk about is transcription control. In transcription control, mechanisms will differ between prokaryotes and eukaryotes. Think about how their DNA works and how big they are and all that. So it functions differently because their structure is different. In prokaryotes, most gene expression control occurs at transcription by transcription factors. These are things that bind to DNA to control speed. And we're going to talk about prokaryotic gene control later, but I will go ahead and just keep going with this for now. Repressors are things that will shut off transcription or slow it down. Think of the word to repress. Operators are silencers specifically to prokaryotes. So there are some things that eukaryotes don't have, but others that they do. And then we have activators that will speed up transition. Think about what it is to activate things. And enhancers, which are activators specific to eukaryotes. So again, eukaryotic cells have their own specific stuff. And something else that can affect the rate of the ability to even transcribe is only unwound chromatin is available for transcription. Remember, chromatin is wound around histones, which are like balls of protein that are stuck together that DNA winds around to make the chromosomes. So if you're wound tightly around that, you can't make your replication fork and be expressed. So factors that can affect the interaction of DNA around histones affect expression. So the methylation of histone proteins can silence DNA transcription. So the more methyl groups you have on you, the tighter you will cling to those histones and through hydrogen bonding, and you will silence your DNA transcription. The other thing you need to know is gene control can happen at mRNA processing, which is post-transcriptional modification. So prokaryotes don't have this, but eukaryotes, remember, they will make their RNA, and then they have to process that RNA. So during this processing phase, the second phase here, you might actually see some modifications that can affect the mRNA might be spliced at different places, or different tails or whatever can happen. So during this step, you can actually affect expression as well. And then the next step is translation, and translation is when that mRNA is read into amino acids. So most eukaryotic control actually occurs at the translation level, which is after the mRNA will leave the nucleus. And one of the things that is affected by or affects the translation rate is how long the mRNA lasts in the cytoplasm, because as soon as that mRNA comes out of the cytoplasm, enzymes find it, they're like, ooh, candy, break it down, let's eat it. So they start breaking down this mRNA because you don't want to spend resources making proteins that you don't need. So the cell has all kinds of ways to keep proteins from being made and only make what's necessary. So if the mRNA is able to stay, quote unquote, alive in the cell longer, in the cytoplasm of the cell, then it will make more proteins, and so that will increase translation. If it gets broken down really fast, then that decreases translation of that mRNA segment. Also, mRNA interference, this is also in prokaryotes, and if you heard me stuttering around on your study guide talking about an RNA not having a complementary strand, this is where it happens, RNA interference. You can have complementary strands of RNA that attach to the RNA and keep it from being read. But that's very rare, but that's where your RNA interference would happen. And then you have D, which is post-translational modification, and that's basically after you have your active protein or your active polypeptide chain. If you see the little scissors here, it can be snipped and changed and modified there as well. All right, let's talk about genes and development. Animal development is orchestrated by cascades of master gene expression in embryos. When I was in high school, we didn't really study this. We didn't really study it in college either, so it was very interesting for me to look at this. I had to, what is a master gene? A master gene is basically this main gene that will cause a domino effect and turn on other genes. So as this one turns on, it will cause all these other things to happen that will cause all these other things to happen that in turn cause these other things to happen. So master genes control like huge parts of embryonic development. As they diffuse through the embryo in gradients, and they can affect expression of other master genes in a domino effect. So there's some overlapping, so gene A turns on and then gene B turns on, but where A and B overlap, there's a gradient there. And so different things happen. It affects segmentation in insects and a bunch of other stuff. So these master genes and their overlap, it really is a big factor in embryonic development. And cells will differentiate according to their exposure to these gradients. So if, like I said, segmentation of insects is the one they use in your textbook, but it's where the overlap occurs is actually where the division of the segment occurs. So the cells that are exposed to that overlap in those genes being turned on will act differently than the ones that aren't exposed to the overlap. So they differentiate or be different things. The expression of homeotic genes during development governs the formation of specific body parts. And this was such a cool section in your text as well, because it was talking about how fruit flies can grow legs on their head because of these homeotic genes. Homeotic genes, in this case, are the leg genes. But they've also found homeotic genes that are kind of like eyeless genes. And this is the next note. It says homeotic genes that function in similar ways in evolutionary distant animals are evidence of shared ancestry. So they did these things called knockouts. So they name genes based on what happens if you make them not able to be expressed. So they took the fruit fly embryos and they caused them this certain gene, they didn't know what it did, but they caused it to not be expressed, and then the fruit fly didn't have eyes, so it became the eyeless gene. So there's all these genes that you could read through in your textbook that are named because of what they didn't, or what they caused to make happen, but they're named in their absence. So the eyeless gene is caused the eyeless gene because if you don't have it, it causes fruit flies to not have eyes, but in humans they have a very similar gene called the PAX6, and it will cause a human eye to not have pupils, which causes you to be basically eyeless or not be able to see. And what they found is if they took this PAX6 gene, mutated it, and put it in the fruit flies, then the fruit flies didn't form eyes. So while we are very, very far distant from fruit flies according to the phylogenetic trees, but the PAX6 gene will do the same thing as the eyeless gene. So there's one gene, side note, that's named cool in German or something because like the guy, after he saw what it did, he was like, wow, that's cool, so it's named cool. So these genes are kind of funny, but they got their name because they would take embryos, especially fruit flies, and they would knock out the ability of the gene to function and figure out what the gene did after the fly developed. So in your next video, we will go in and we'll talk about gene control specifically in eukaryotes.